Every cell in our body carries a story written in DNA. On this World DNA Day, we celebrate the molecule that defines life, identity, and innovation. World DNA Day is celebrated every year on April 25th, marking two landmark events in the history of science: the discovery of the double helix structure of DNA in 1953 and the completion of the Human Genome Project in 2003. These breakthroughs transformed our understanding of life, opening the door to modern genetics, biotechnology, and personalized medicine. This day is not only a celebration of scientific discovery, but also an opportunity to recognize how DNA research continues to shape our world. From diagnosing diseases and developing targeted treatments to tracking biodiversity and understanding the spread of antimicrobial resistance, DNA plays a vital role in addressing some of today's most pressing global challenges. The World DNA Day reminds us of the power of science to uncover the unseen, to connect all living organisms, and to drive innovation for a healthier and more sustainable future. As the Molecular Microbiology and Human Diseases Project at the National Institute of Fundamental Studies in Kandy, Sri Lanka, our research explores DNA across diverse fields. Our DNA-based research spans across human health, environmental science, and microbial ecology, addressing some of the most pressing challenges of our time. This is driven by a main goal to understand how microorganisms interact with humans and the environment, and how genetic information shapes these interactions.
In our lab, using advanced DNA-based techniques, we investigate microorganisms attached to microplastics collected from multiple locations across the Kandy Lake, Sri Lanka, including inlets, mid-lake regions, and the main outlet. Our findings reveal that microplastics are present in all the sampling sites and act as hotspots for microbial colonization. Through DNA analysis, we also detect antimicrobial resistant genes associated with these microorganisms, highlighting how microplastics can act as vectors for the spread of antimicrobial resistance in aquatic environments. By linking DNA-based microbial data with environmental factors like temperature and air quality, we examine how airborne microorganisms influence respiratory health and disease patterns. Using molecular techniques, we extract and analyze DNA from airborne microbial communities to identify the diversity and the abundance of bacteria present under the different environmental conditions. This allows us to detect shifts in microbial populations at the genetic level, including the presence of potential pathogens and antimicrobial resistant genes. At the Guhagoda dump site, Kandy, Sri Lanka, our research focuses on the DNA of airborne microorganisms and microplastics. Using molecular techniques, we extract and analyze microbial DNA from air samples to identify species and detect antibiotic-resistant genes. We specifically study how microplastics act as carriers of these microorganisms and their genetic material, forming what is known as the plastisphere.
We investigate how antibiotic-resistant bacteria emerge and spread in unique environments like hot spring. By identifying resistant genes, we gain insights into how microbes adapt and transfer resistance that is critical in combating the global threat of antimicrobial resistance. Through transcriptomic analysis, we study gene expression patterns in diseases such as tuberculosis. By analyzing DNA and RNA from patient samples, we aim to identify reliable biomarkers that can help to distinguish between latent and active infections, contributing to earlier diagnosis and improved treatment strategies. Our work also extends to study about toxin-producing cyanobacteria and also study genes responsible for toxin production, such as the microcystin gene cluster. This research helps us to understand how genetic variations influence toxin production, which is essential for ensuring water safety and protecting public health. Through these diverse projects, DNA serves as a powerful tool allowing us to detect, understand, and respond to complex biological systems. As we celebrate World DNA Day, let us contribute to explore, innovate, and harness the power of DNA to shape a better tomorrow.